Hello, class. Hope you guys are doing well today. Had a great Halloween and a great weekend. So today we're going to go over soils. Soils as a reservoir, carbon reservoir. Um, so let's get started. So first of all, why are they important? Well, first they're kind of a larger reservoir compared to other ones. They're not the largest of all reservoirs. If you look here, soil's got about 1,500 units. Definitely not as much as the rocks or the deep ocean, but more than vegetation, almost 2 times more, or 2.5 times more, about 2 times more than surface water. It's got more than sediments or biota or wetland soils. So there's quite a bit of carbon stored within our soil. Now, soils are more actively engaged in interacting with the rest of the carbon cycle. So what that means is that changes in the soil carbon pool can have a large effect on the atmospheric CO2. So that's good, right? So we want to know a little bit more about it. So what are the factors that actually control the inputs and outputs of the carbon within the soil? So this map here is actually going to be a map of SOC, or soil organic carbon. So if you look, basically it's going to be a measure of what's growing. Sometimes you can look at it as kind of a measure of photosynthesis. Where things are not growing, or there's very little photosynthesis happening, like in the desert, there's not very low values of SOC. But where things are growing a lot, where we have quite a bit of photosynthesis going on, then you're going to have these higher levels. So the input to this, though, is going to be the NPP. So going back to last week, NPP is going to be, after we account for plants respiring, everything that's left in the plant. Sorry. Okay, so these things right here are going to be the things that affect the amount of soil made after, everything's left in the plant. So the things that are going to affect it are going to be the climate, the amount of CO2, site fertility, hydrology, what's going on with the water cycle in that area, and the composition of the different species in the area. So once we have, the soil carbon, it's either going to go up into the atmosphere through CO2 and methane, or it's going to go down in rivers and streams, and we're going to have like dissolved organic carbon formed. The temperature, the water, oxygen, the quality of the substrate, any type of fires, that's going to be the things that are going to determine whether or not it goes up into the atmosphere or down into the rivers and the streams. Okay, so when we're talking about carbon storage, we're really in soil, we're really talking about decomposition. So the factors that can affect decomposition are the things like the organic composition, which brings us to these two terms here. So the first term here is libel. Libel is going to be when when carbon is recycled a lot in the system. It's because whatever is going on is very attractive to the bacteria. Okay, so it really wants to eat that soil that's going on, that we have there. And the other term is going to be refractory. This is where, refractory is not interacting very much. It just pretty much hangs out. So whether or not something is libel or refractory is determined by the composition of the soil, and that will determine the rate of decomposition. And so another thing is going to be the size of the actual molecule. Another one will be the level of protection. And then we also have some climate controls here. These are all things we'll go over here right now. All right, so different compounds decompose at different rates depending upon their organic composition. So this graph here, on the y-axis, we have the mass remaining. So the percentage of the original. So we have 100% here, the original thing, which I'm just going to kind of, for explanation purposes, I'm going to say something like a branch that might fall out of a tree, or leaves right now in the fall, we have a lot of leaves falling. So it's a good time to kind of think about that. On the x-axis here, we have time. And you can see that things are a lot faster. Things decompose a lot faster in the tropics than they do in the Arctic. So we have the whole composition of the leaf or whatever we're looking at here. And you can see that there's a very fast decline. Like it's a very fast at how much is remaining. And then at some point here, it just kind of levels out and it just kind of like stops declining as fast. So what's happening here is the things that are more attractive to the bacteria that it likes the most, it's going to consume these the fastest and immediately. And so it's going to have that steep decline of these things here. Now, what's left in the long term, though, are going to be the microbial products. And this is things like, the cell of the bacteria. It has to basically sustain itself. And the other one is going to be lignin. Now, this is basically going to be like starchy molecules. So think about like a corn husk. This is going to be something that's harder for the bacteria to digest. And so they just slows down the rate of decomposition. It's not as attractive to the bacteria. So it's going to be there for a longer period of time. Okay, and then another thing that will actually affect it is going to be the stability of the soil. So when we're talking about the stability, we're talking about mostly refractory. So the things are basically just kind of hanging out there and not doing much. That's when we have a stable soil. So the things that are refractory are going to be aromatic and aliphatic compounds, which are going to be wax, like wax examples are waxes and lipids. So if we look at the waxes, this is going to be like this ring-like structure here. These are kind of harder for it to break down. And then also the long chain-like structures here, like hydrocarbon. These are also going to be harder for it to break down. So these two are examples of the refractory molecules. But but we also have the other side, which is the labile molecules, and this is the sugars and the amino acids. These things are very attractive to bacteria. They really like them. They're necessary for them to survive. So they're definitely going to want to eat these first. So recalcitrance leads to selective preservation compared to easily degradable material. So basically what we're saying here is that we end up selectively preserving the refractory molecules. And that leads then to a stable organic carbon. And it can leave it there for a few decades. Another factor influencing lability is going to be the size of the molecule. Now, we're talking about like single cell bacteria here. If it's too large, it can't get it inside of its cell. And when that happens, it has to use these things called exoenzymes in order to start breaking it down outside of the cell so then it can bring it into the cell. What this basically means is it's harder for the bacteria to break it down. And since it's going to be harder for it to do that, then it's really going to probably leave the larger ones alone. The type of the chemical bonds is going to be another one. Remember those aromatic ring-like structures, those like these right here, these are going to be harder for it to break down. Toxicity, there's actually things that are toxic to bacteria, so they kind of like leave them alone. And then another thing will be nutrients. Like I said, sugars and nitrogen and phosphorus, these are going to go fast. They're going to be the first things that go because it's necessary for the bacteria to live. The other things left over will be a lot slower. Another thing that can cause it to be stable is when organic matter gets stuck onto clay particles. So, I think I mentioned organic matter, can get stuck, or the organic matter and minerals become glued. So you can see here, there's the organic matter and here's a clay particle. When they become stuck to each other, then the organic matter is not going to get broken down as easily, and so it's going to become more stable. And then the last thing here is when things actually destabilize the soil. The first one's going to be soil management, because sometimes you actually want the bacteria to break down the soil. This happens more in agricultural purposes. Also changes in pH, because it's very sensitive to pH. It affects the saturation state. That basically is going to change where you are in the equilibrium for some reactions to happen. And so whenever the pH changes, then it can make it less stable. We also have the inputs of easily degradable organic matter, or what they call the priming effect. It's kind of like if you have something that's super yummy next to something or like attractive to the bacteria and it's already kind of eating, it might continue eating something that's a little bit more difficult or less appeasing to it. And then the other one is going to be, climate factors. So things like dry, wetting, freezing, thawing, those kinds of ideas are going to make it, not as stable. So this is everything I have for you guys today as far as lecture. Please make sure that you are reading chapter 4 in Archer. I also put up something different, which is a Spark page. I hope you guys get a little bit have some fun with it and get a little artistic, creative, I guess. So I'm kind of looking forward to seeing what you guys actually come up with. And, let me know if you guys have any questions.